So our final speaker of the morning is Stephanie Cherqui, who will tell us about her efforts to truly treat and perhaps cure people with terrible disease, cystinosis. Stephanie.
Thank you very much, Larry, and thank you for the invitation. We are still at the clinical stage of this approach, and I want to talk about the hematopoietic stem cell gene therapy. So I will first present the disease because I'm sure most of you don't know it. Don't know how. Oh, it's this one. Right. So it's a rare disorder, and the gene involved is called CTNS, and it's ubiquitously expressed. It encodes a transmembrane protein located in the lysosome, and the function of this protein is to allow the cysteine to exit the lysosome and then to be reduced into cysteine. So in patients, the mutation or deletion of this gene results in the dysfunctional protein, and the cysteine stays stuck in the lysosome and create these characteristic cysteine crystals. And because the gene is expressed in every single tissue, then we end up with a multisystemic disorder. So first, they have a renal Fanconi syndrome. They cannot retain any kind of water or nutrients. They end up to end-stage renal failure, so they have to have a kidney transplantation. But also, then they develop a multi-systemic disorder, as I said, including blindness, photophobia, diabetes, myopathy, and neurological disorder, among many others. So the treatment that the patient has so far is first supportive. So the kids have to take up to 40 pills a day to be able to try to treat every single symptoms. But there is also, fortunately, the drug called cysteamine that allows the exit of cysteine out of the lysosome. But unfortunately, this drug has multiple severe side effects, but most of all, it's only delaying the progression of the disorder. So the patients still die when they are young adults. So the approach that I always wanted to take is gene therapy, but unfortunately, this was a very challenging disease to do gene therapy on because the gene is expressed everywhere and all the tissues are degenerating. So I was thinking then we need a vehicle to bring a healthy gene in every single tissue. And what better a vehicle than what we all have in our bone marrow, so the bone marrow stem cells. So to make a long story short, because I don't have that much of time, I will just present our data on the hematopoietic stem cells, which we are lucky worked really well. So we use a mouse model of cystinosis that replicate very well the disorder with cysteine accumulation and tissue degradation. And what we do is we first did a proof of concept using a GFP transgenic mice, so to be able to follow the cells after transplantation. So we do a lethal irradiation of the mice to wipe up their bone marrow stem cells, and we do a simple tail vein injection in the mice, so the cells then repopulate the bone marrow and stay there for the life of the mice. So what we have seen is, this was only 4 months post-transplantation, and that was very surprising, is that in every single tissue of these mice, there was an abundant amount of bone marrow-derived cells, so green cells in every single tissue, including kidney. So this means that because of a tissue injury underlying cystinosis, the cells were called to treat and repair the tissue, which is the function of the bone marrow stem cells. And this correlated with a dramatic decrease of the total cysteine amount measured in the tissues. So these are the mice transplanted with CTNS deficient hematopoietic stem cells, and you can see that between 2 and 4 months, you have an increase of cysteine in every single tissue. Whereas the mice treated with wild-type hematopoietic stem cells, you have a dramatic decrease, and this can go up to 90% of clearance of the total amount of cysteine. So the next question is that, is this stable for the life of the mice, first of all, and can this result to the tissue preservation? So we analyze the kidney because it's the tissue primarily impacted by the disorder. And you can see here that at the 15 to 17 months old mice, so they were transplanted at about 2 months of age. So 1 transplant, and we check them at the end of their life. And you can see that in non-treated mice, the tissue is completely degenerated and the renal function is impaired. And this is a representative picture of one of the treated mice. So 1 transplant at 2 months and 1 at the end of their life, you have just focal anomalies in the tissue, and the kidney function is completely preserved. So that was really great. But we also have some mice that, even if they were treated, were like not have a good kidney preservation. And what we realize is that the quantity of cells expressing the CTNS gene into the bone marrow matters. And this is a very important point when we will want to do the patient. So we have to make sure that we reach a good threshold. So more than 50% of the cells must express the CTNS gene to have a good tissue preservation. So then also at this age, we have abundant cystine crystals, and in the treated mice, there is almost no more cystine crystals seen in the kidney. And this shows that the cells don't go randomly in the tissue, but they really do their job, which is releasing the cystine and cystine crystals in the tissues. The second tissue that were mainly impacted in cystinosis is the eye. And there is an abundant accumulation of cystine crystals within the cornea. And this results into opacity of the eye. In the mice, we replicate that very well, and because we don't treat them, we see a complete degeneration of the eye. The treatment is a cysteamine eye drop that the patient have to put every single hour, so the compliance is very poor. So what we have done, this is, so usually it's a movie, but it's not working. So this is a non-treated mice, and after 1 year of post-transplantation, we measured by a confocal macroscopy the cystine crystals in the eye of the treated mice. And as you can see, this is much better. And if we can see that in the transversal section, this is the mice that with a high level of engraftment of the cells with expressing CTNS. And you can see that the release of cystine crystals is up to 90% in the eye. We also showed that we could rescue the eye structure. So this is very great. 1 transplant, and for the life of the mice, we can rescue not only the kidney, the eye. We have also showed the same thing for the thyroid, and so on. So now the question I had often is, how does it work? How can hematopoietic stem cells can allow long-term tissue preservation? And so the first question was, do the cells differentiate, fuse, or transdifferentiate? So I generated we generated in my lab a new mouse model which was red, so expressing a DS red reporter gene everywhere, and we transplanted the green mice, the green cells. So I was hoping, and I was very sure to see plenty of yellow cells everywhere, thinking that it would be fusion. But unfortunately, so we had some, so there is some kind of fusion, but the most of the cells were still green, showing that it's not a fusion, and actually they are differentiating to macrophages. So that was a very big surprise, because I was wondering how can macrophages can result in a long-term preservation of tissue. So of course, they have their capacity of keeping a good homeostasis of a tissue by phagocytosis, but the other hypothesis that came up was a cross-correction. And cross-correction has been well shown for secreted enzyme, but it has never been shown when the protein is a transmembrane lysosomal protein. So we, to make a long story short, we showed that cross-correction in the context of cystinosis could occur. So when we co-culture macrophage with CTNS deficient DS red fibroblasts, we sort them and measure the cystine content, we could have a dramatic decrease of cystine and what we showed is that cell-cell contact was necessary. Macrovesicles can lead to decrease, but is not sufficient. And what was surprising is that in our Petri dish, we were seeing a lot of this long tubular plasma membrane protrusion. And so we did some literature search, and actually these protrusions were described in 2004, and they are called tunneling nanotubes. These tunneling nanotubes are shown to be important for intercellular communication, and they also mediate pathogen spreading. So to see if our cystinosine could be transferred via these nanotubes, we stably transduce the macrophages with a lentivirus vector expressing the fusion protein cystinosine GFP, and we co-culture them with a fibroblast. And unfortunately, my movie didn't work, but we really see the vesicle containing cystinosine going from the macrophages to the deficient fibroblast. And actually, we describe this vesicle, and they are the lysosome. So what's happening is that the macrophage can create this protrusion and deliver the healthy lysosome to the disease cells. And what we showed also is that it's a bidirectional transfer, because even the disease lysosome, containing the cystine, can go and via the same tunneling nanotube and go to the macrophages. So this was in the plate. I wanted to make sure that this was true in vivo. So we studied the kidney again, and we showed that the macrophage, for example, for the one who know how a renal tubules looks like, it's epithelial cells that are surrounded by a very dense tubular membrane. And we showed that the macrophage were always surrounding the tubules, but never go inside. And what actually, oh, yeah, but we don't see that's too bad. It's very beautiful. We show that we have a really nice movie, and I can show for people who are interested on my computer that the tubes, they generate the tube and that cross the tubular basement membrane, and so they cross the membrane. But also we showed that using the no-color mice and DS-red hematopoietic stem cells transduced with the cystinosine GFP, that not only they cross this membrane, but they can deliver the healthy and cystinosine-containing lysosome into these epithelial cells. And that's how we can explain this long-term rescue of tissues. So this was for mechanism of action. But now what we are interested on is how do we go from bench to bedside. So this work led to the first stem cell clinical trial for cystinosis, which consists in allogenic hematopoietic stem cell transplantation. And we decided with Don Cohn and Ted Moore to use healthy sibling to reduce the risk of this strategy, which is very risky. So this was just to test the efficacy and toxicity of hematopoietic stem cell transplantation in cystinosis. But we wanted to have a 10 out of 10 HLA matched sibling donor, again to reduce the risk. And this requirement, all the candidates we had didn't match this requirement. Now we have a candidate that matches this requirement, that would like to enroll, but we are so close to the autologous strategy that I will talk about now, that we will probably have this patient just go with a first trial that we are developing now, and that I'm talking about here. So what we want to do is to do an autologous stem cell gene therapy strategy, meaning that what we want to isolate the stem cells, so just in the peripheral blood from the patient, ex vivo modify them using a lentivirus vector containing the CTNS cDNA. And so not all the cells will express CTNS, but hopefully most of them. And the patient will go through myeloablation to remove their own stem cells, and the stem cell will be retransplanted in the patient. So this approach is much safer. The risk, we are now below 5% of mortality or morbidity. The problem is that it's the quantity of genes that are introduced. It can be an issue. We need to make sure we have the right protocol to target the most cells we can. And there is also a risk of integration in oncogene. But so far, I have to say that with a lentivirus vector, there is no major adverse event that have been seen in the many clinical trials that are going on now, and using the same approach. And Dr. Cohn, who will be our keynote speaker this afternoon, will talk about his own clinical trial using a lentivirus vector that he gave us, and this is the same one so the same backbone we are using, and we subclone CTNS cDNA in this backbone. And now what we've done, so of course we did the preclinical study with the mice, showed that the cysteine decreased, the kidney function improved, and the cysteine crystals are significantly decreased. So this approach works as well. And we then contacted the FDA and now our clinical product will be the stem cell genetically modified with a lentivirus vector. We had a pre-IND in 2013, and they agreed, and we discussed about the best pharmacotoxicology they wanted because it's considered like a new drug, so we have to do everything like a new drug to put a new drug on the market. And we had to buy a GMP comparable lentivirus preparation from Ken Cornetta, and we had to put in place GLP lab facility at UCSD, which was not an easy task, but now we have them. Now we are in the middle of a pharmacotoxicology study, so for a matter of time, I will just go quickly with that. We just target a vector copy number of between 1 and 3, so we want 1 and 3 vector per cell. We did the colony forming assay, so I will just go fast on what the FDA required to approve our strategy. So this is, we make the cells differentiate in vitro, so we compare the transduced and non-transduced. And we had no skewing on the number of cells or phenotype. So here you see the vector copy number we obtained in the healthy donor, which was right in the range of 1 to 3. And you can see that in the patient, we are just below 1. And so this is, I think, not enough. So we are still trying to improve our protocol to increase this number, because I think we really need to, and we saw that in the mice, we need to be really between 1 and 3 to get the therapeutic effect. So, of course, we will have to, we studied the vector integration site, and this is Frederick Bushman at University of Pennsylvania who does that, and he didn't see any weird distribution of our vector in patient cells. The other in vitro assay that the FDA requires is the in vitro immortalization assay, which is a large expansion of the cells when they are transduced compared to control. And this was done in Cincinnati. There were no immortalized clones. So again, the safety profile is good. So all this will be integrated in our IND. And the last portion is the in vivo toxicology studies. So we have to we have primary and take the bone marrow of the primary recipient to put in the secondary recipient, and we have a tremendous amount of tests to do in the mice, which costs a lot of money and takes a lot of time. But we are we are well advanced, and we think that we have like a few months left into these studies. So this is where we are. We are almost done with the pharmacotoxicology study. We need to now apply to IND, IRB, RAC, and we to find the funding. We will have then be able to recruit patients, and we will enroll 6 patients total. So I put together a consortium with 13 members. Most of them are physicians who will participate to this clinical trial. And the study design is, we will mobilize the cells using GCSF and PRC4. The ex vivo transduction will be done at UCLA and using a GMP virus preparation. We will have a full myeloablation of the patient, and it will be a one-time intravenous infusion of the cells. So we will do also a staggered design. So what we want to do is to first enroll 2 patients the first year, and do this study and learn from that to maybe modify or optimize the protocol for the two next. And if a risk assessment is good, we hope to be able to do 2 adolescents for the last stage. So this clinical trial will be very complicated because we have many tissue to look for. So first we will have Dr. Ball at Moore Cancer Center who will isolate the peripheral blood stem cells. The apheresis product will be sent to the GMP facility directed by Donald Cohn at UCLA. It will isolate the CD34 cells and transduce our patient cells. Then the frozen product will be sent back to UCSD, where they will be transplanted back to the patient. And before, because we want also to show efficacy, so not only safety, but we would like to show efficacy. So we will, before and after transplantation, at different time points post-transplantation, the patient will go through a lot of clinical studies, including kidney study, of course, blood work. And we will also measure the cystine content, crystal content in the tissues by rectal biopsy. And we are also optimizing a confocal approach with using a skin confocal. And I will test that at the grand opening of the cystinosis facility clinic at UCSD. So the eye, and we do the CT, plenty of other things. And so I just want to make sure that I acknowledge the right people here, because this is very complicated, and I would have never been able to put that together without the help of the Sanford Stem Cell Clinical Center and the CERM Alpha Clinic that really help us every step of the way. I want to acknowledge everybody in my lab who did a tremendous amount of work, all our collaborators, Donald Cohn, of course, the consortium member. Again, all the members of the Alpha Clinic, they have been very helpful. I want to acknowledge the funding, and also I want to say a big thank you to the grant we just received from the Sanford Stem Cell Clinical Center, who will not only allow us to apply to all the regulatory agencies, but who will also cover the cost for the 2 first patient transplantations. So this is our first funding for the beginning of this clinical trial. So thank you.
Another collaboration on the mesa. Do you need to do the full panel with the sitting down? No, okay, all right. So, and I'm glad to see that Stephanie is using a jet aeroplane rather than pushing a boulder up a hill.
Given the patients will be receiving their own marrow, I wonder why it's necessary to ablate their marrow in advance of the treatment.
So if you don't ablate the marrow, the stem cell don't engraft in the bone marrow, and we did that in the mice. The efficiency is very low, is very poor. The donor transplanted cells are almost not seen in the blood then, and so we don't have any effect.
Hi. You elegantly showed in your mouse model that hematopoietic stem cells, presumably through cell-to-cell contact, correct the defect. And then you mentioned your clinical trial of matched sibling donor transplant. You did not tell us the outcome of that.
So, as I said, I mean, I went too fast probably. It's that it was very hard. We had a lot of candidates that we tested for their match, because they have to be 10 out of 10 match with a matched sibling donor, healthy sibling donor. And all our candidates were not, and we just have one now. But as I said, it took so long to find this candidate, and now we are so close to the autologous transplant. I mean, we discussed with Don Cohn and all the members. I think we think that it's much safer to go to the autologous transplant directly than to go to the allogenic transplant, except if the FDA or the IRB require it. In this case, we will have to go through that.
It seems to me that that's a new paradigm, because every instance of hematopoietic stem cell gene therapy has been preceded by demonstration that hematopoietic stem cell transplantation benefits the patient. So I think that that's a step that I would like to see before we go to the gene therapy.
And we were sure about that, actually. We were sure this is what the FDA wanted to see. And that's why we opened this clinical trial, a few years ago. Actually, the FDA didn't require it. So, and why? I think it's because there are other clinical trials out there. So a lot of them is for blood disorder, using this approach. But there are some already using this approach for lysosomal storage disorder. And the outcome is really good. So far, it's really good. So I think that's why the FDA maybe and the IRB might not require it. But we cannot be sure at 100%.
We're going to have to wrap up here. Sorry. We're scheduled for a break, so let me just thank the panelists and speakers from the previous session. I think you've gotten a little snap.